Okay, let's get started. Welcome to Medicine Grand Rounds. It's good to see you, and I look forward to your questions and comments, which you can put in the either the chat box or the Q&A box. We'll get to all of your questions during the discussion period. Today we have two speakers that are going to focus on population genomics. Dr. Chris Gignoux is a professor of medicine in the Division of Biomedical Informatics and Personalized Medicine here at the University of Colorado. He received his bachelor's, master's, and PhD from Stanford University and did a postdoctoral fellowship in human genetics at Stanford. He's a population geneticist that focuses on genetic ancestry to understand the distribution of disease in populations, and is an ideal faculty member of biomedical informatics and personalized medicine, and a major contributor to the Center for Personalized Medicine here at the University of Colorado. Kathleen Barnes is a professor of medicine, and Dr. Barnes serves as the director of the Colorado Center for Personalized Medicine and is the division head for biomedical informatics and personalized medicine. Dr. Barnes received her undergraduate degree in nursing from the Medical College of Virginia and a PhD in anthropology from the University of Florida. She then did a postdoctoral fellowship at Johns Hopkins University and studied with Dr. David Marsh, focusing on asthma genetics. She established her career at Johns Hopkins University and rose through the ranks of professor of medicine, and focused on the influence of ancestry on the genetics of asthma. We were very fortunate to recruit her to the University of Colorado about 5 years ago to lead the Division of Biomedical Informatics and to build the Colorado Center for Personalized Medicine. She's done a spectacular job at both of these endeavors, developing an incredible infrastructure that you're going to hear more about, and recruiting talented faculty to the Division of Biomedical Informatics and Personalized Medicine, including Dr. Gignoux. So I think Kathleen is the first speaker, and then Chris will follow her.
Thanks so much for that introduction, David. Really appreciate it. I'm going to start off with our case report, and this slide describes this. So this particular patient was a 77-year-old white male with a past medical history that was significant for AFib, hypertension, type 2 diabetes, listed on the slide quite a bit. He was status post a lateral MI requiring a stent placement in 1994. And among the meds that he was on following this event was clopidogrel. We're fortunate that this patient was a Colorado Center for Personalized Medicine, or CCPM, Biobank participant. And what that meant for him is that we had performed genotyping in the gene CYP2C19. And we were able to provide pharmacogenetic results back into his electronic medical record, back directly to Epic, indicating that this individual's CYP2C19 phenotype star2, star17 makes him an intermediate metabolizer for clopidogrel. The patient requests a refill for his clopidogrel, but because this clinical decision support was built around this particular drug gene pair and in Epic, an alert fired to the provider, and the refill was denied and an alternate therapy was prescribed. Now, one fun, interesting fact about this particular patient is that he was actually also a retired cardiac surgeon. So he really wanted to take control of his own health. What you see in this screenshot is really how it works, what we've built out and how it works. So this is a screenshot of the best practice advisory, or the BPA, that's in our system. So you can see that the genetic information for this particular patient had been added on May 23rd, 2019. And it summarized that this patient's an intermediate metabolizer for clopidogrel, given his phenotype, and the BPA suggests that he should be prescribed ticagrelor or prasugrel as an alternate to clopidogrel. So as a follow-up to this, a few months later, the patient sent a message to his provider saying that he had experienced slow, progressive, superficial bleeding since taking ticagrelor, and he thinks it might be too effective, and he requested to switch back to clopidogrel. The provider agreed that the risk-benefit ratio favored clopidogrel, and this change was made. And interestingly, at that second point in time, there was another alert firing saying that it wasn't recommended for this patient to receive clopidogrel, but again, the provider and the patient made a calculated risk together. And the provider in this case argues this is the ultimate example of personalized medicine. So to summarize this case report then, the medication change was made with significant potential benefit based on the PGX information, the preemptive genetic information that we had for this patient that was facilitated by our clinical decision support tools. The phenotype prediction was most likely accurate based on his physiologic response to his alternate therapy. And ultimately, he's able to make a more informed decision based on more complete or personalized information. So with this, I want to introduce to you, if you don't already know, about the Colorado Center for Personalized Medicine and what we've built over the first 5 years. The first half of this presentation, as you've already gathered, is going to really focus on our development around pharmacogenetics, or PGX. And then Dr. Gignoux is going to segue into the focus on the role of ancestry, which is of considerable interest in our program. So to start with, as David already mentioned, one of the first things we needed to do upon building the program, that was 5.5 years ago, was to create an academic home for the faculty that we needed for this. And this is the Biomedical Informatics and Personalized Medicine Division. The health data warehouse called Compass, or Health Data Compass, had actually been established the year before I arrived. But we were able to expand that, and we're proud of the fact that Health Data Compass actually represents a banner partnership with Google Cloud, where ultimately we've been able to move all of these data to the cloud. We needed a high-performing performance computing cluster to manage all of the anticipated data. We established this. Caveat here, I won't spend time on it, but in the second 5 years, which we've entered into this year, we're now moving all of our data to the cloud. And so that has reduced our dependency on an on-prem infrastructure. I won't go into a lot of detail for the sake of time, but I've presented before, and you'll hear bits and pieces about it. We made a decision on what sort of a genotyping or genetic platform that we wanted for this program. We elected a high-density SNP chip, which actually Chris and I had previously co-developed in partnership with Illumina before either of us moving out here. It's been incredibly enriched for clinically valuable data to be able to return genetic test results. Most importantly, we were able to establish the CCPM Biobank, and we made a decision that we've never regretted to make this a CLIA and ultimately CAP certified biobank, and you'll hear why that's important. In very close partnership with UC Health, which have been incredible champions for the program, we were able to launch an electronic consent through the patient portal, My Health Connection, and I'll share a little bit about that. And then ultimately, we've been able to launch the return of pharmacogenetic results, as you've just seen from this case report. We're now moving into a phase to build out a process and a pipeline for returning other types of genetic results besides PGX. And unfortunately, I won't be able to go into that today. This is what the center looks like. We think about starting at the top, if you will, with the UC Health adult patient population, close to 2 million active patients in the UC Health system, which of course cuts across the state and region. I've mentioned the biobank. We have, as of yesterday, over 173,000 participants enrolled in the program in the first 5 years. I've mentioned this particular SNP chip, Illumina's Mega EX chip that we've used for genotyping. We've generated genetic data on the first 32,000 to date. On the other side, of course, importantly, is all of the rich phenotype data available in the EHR. That is all brought into our health data warehouse or health data compass with right around 9 million patient records to date. And all of this gets brought together in the biocomputing and analytical phase. Several points that we're especially proud of over the first year, which we think makes CCPM a differentiator, certainly nationally compared to other programs. Again, we're one of the first enterprise health data warehouses on a public cloud. Our CLIA Biobank is one of the only of its kind nationally, and you'll again hear why that's important. We're actually the first personalized medicine biobank to leverage a commercial SNP chip, if you will, for return of clinically actionable results. We're one of the first programs to launch clinical integration of PGX via a research biobank. And important for everyone to know is that we're currently scaling up our recruitment with a very lofty goal of 100,000 participants per year by the end of the next 5 years. Part of the impetus for this scale up, and again, an incredible close partnership with UC Health, is an agreement that we signed just under a year ago with Regeneron, which is performing whole exome sequencing for all of the samples that we send to them, sending the data back to us. And through our clinical biobank, the data will be clinically validated so that ultimately we can return clinically actionable results to our patient population. So how does it work? This is an image of the workflow, if you will, in terms of recruiting patients into the program. As I mentioned, they see the opportunity to consent to be part of the biobanking initiative through the My Health Connection portal. That's step number one. Step number two, excuse me. Sorry about that. Step number two, the next time that they come in with an order for a blood draw, Epic will trigger the order for a dedicated blood sample that will come directly to our biobank. And so we always have a ratio of more consented to blood samples because an individual in ophthalmology, for example, may consent but not have his or her blood drawn for some time. After the blood is collected, the DNA is extracted in the biobank. And then as you'll hear, and I'll go into some detail, that genetic data is generated and ultimately allows us to return clinically actionable results. What makes us unique is that we're able to maintain clinical custody of the biospecimen from the time it's collected while it's being processed in the biobank. And we've gone through the process of technically and analytically validating content so that we can directly return to our participants. This is the biobank enrollment to date. As I just mentioned, we're just over 173,000 participants so far. You see again that 1-to-3 ratio of patients to samples collected. I mentioned already that the first 32,000 have been genotyped. And this shows you how many samples based on the version of the consent that was used are eligible for research. And you're going to hear a little bit more about that from Gignoux. Importantly, we have quite a large footprint, not just in the state and region, but even nationally. And this is a recent map that Dr. Laura Wiley and our team was able to generate, demonstrating that representation of our biobank nationally. A quick screenshots of what it looks like. If you haven't been in yourself, I encourage you to do so. If you go into your My Health Connection, this is what the UC Health Epic team has built for us. And so you see this opportunity to enroll directly into the biobank. It will alert the patient that he or she has consents that need to be reviewed. That takes them to the CCPM biobank consent. They have the opportunity to look at a video which explains more about the process, what is DNA, what is personalized medicine, and so on. And they can electronically consent at that particular point in time, or they can decline. And if they decline, we have a policy of not putting this in front of them for some period of time thereafter. What's unique about our consent and important for the research community to be aware of is that part of the consent is that a participant agrees to be recontacted for other types of research. They agree that the data can be shared, not just in places like dbGaP, but with our potential industry partners such as Regeneron, And they agree to receive genetic test results. Now, at this point for PGX, that allows us to directly return those data into their medical record. For the more sensitive type of genetic results, the pipeline we're building will provide an additional opportunity for the patient to tell us that he or she does not want that type of information. And really importantly, and this has made such a difference for us, is that consent triggers, as I mentioned, an automated blood collection or blood order for a dedicated tube. So how do we stack up with other personalized medicine programs, many of which have been around for some time? And so this was a review that came out now almost 2 years ago. Close collaborators of ours at the Mount Sinai BioMe program, Emer Kenny and Nora Abul Hussein. This was a review that appeared in Cell and a figure from that. And it was really focusing on ancestral diversity or lack thereof across biobanks, both nationally and internationally. You see here one of the oldest biobanking initiatives, for example, Decode Genetics in Iceland, a program in Estonia, and so on. Several of the Asian biobanks represented in red. And the green circles here indicate the more diverse biobanks out there, and showing you where Colorado Center for Personalized Medicine, how we stack up both in terms of the age of our program and the percent of non-European ancestry in our biobank. And you'll hear more about this from Chris in just a few minutes. A couple important points to remind everybody in how we build a CLIA certified biobank laboratory and what the criterion are. Samples, as I mentioned, as they come in, DNA is extracted, and we genotype them to date on Illumina's megachip. I've already mentioned and shared with you that we have a CLIA certified biobank and why that's so important. And what you see over here on the right are the CLIA CAP validation metrics, which we adhere to very closely. And I have to shout out to Christy Crooks, who's the director of the CCPM Biobank, and maintaining this level of accuracy within the biobank. So when we made the decision to move forward with the PGX initiative, a lot of things had to happen. And first and foremost, I want to underscore that none of this would have happened without the leadership of these two individuals, Dr. Chris Apolante, who holds a joint appointment with BIPM and CCPM, and has been directing this program in conjunction with our CCPM medical director, Dr. Dave Kao. So one of the first things that the team did was put together a committee called the Pharmacogenomics Implementation Committee Colorado, or PICCOLO. And two of the major challenges for this group was, first of all, to construct the technical and the clinical informatics infrastructure, whereby clinical PGX results could be returned preemptively to the EHR, as you saw with the case report. The second big challenge is to build the clinical decision support, or CDS, tools to inform providers when they prescribe a PGX medication to a participant with an at-risk genotype. So the first step was to form this working group. As I already mentioned, that was launched in August 2016. And on the right side of the slide, and I won't read this out, but basically what this summarizes are the four major steps that are involved in the systematic drug-gene pair development process, which includes evidence review, multi-level stakeholder engagement, again, to build and test that CDS solution, and really importantly, education and communication. And we have a number of folks in the program who lead these various initiatives. One of the things I also want to mention with this slide up here is that we use several types of evidence to base our drug gene pair development processes. These include national sources or international sources, but including Pharm, GKB, CPIC, and also FDA labels. So this slide represents CCPM's PGX CDS development to date. We started with one gene, and the example that I gave you, CYP2C19. And our rationale for selecting the drug gene pairs has a lot to do with what's available to us on the genotyping chip that we use, what content can be reliably, technically, and analytically validated. We have not prioritized these rollouts in terms of prioritizing any particular clinical trait or disease, as you can see from this slide. So for CYP2C19, which I mentioned, you see a number of drugs that are associated with variants in this particular gene. We use an interruptive alert or non-interruptive alerts that are in line with medication warnings. You can see from here to date, we have warnings of just under 70 unique patients of those first 32,000 that have been genotyped. The different types of drugs are listed here. We're also working on return of results for a number of new genes and drug gene pairs that are available, including DPYD. And we're particularly excited about this one because DPYD, patients with DPYD variants may not be able to metabolize certain chemotherapeutic drugs at a normal rate, and they're therefore at risk for potentially life-threatening toxicities. They're not routinely tested for in clinical practice, and therefore the ability to provide this information to our patient population or our providers through the Biobank initiative would really enable more personalized care in this space. A final important thought here is that about 3 to 5% of European ancestry individuals have partial DPD deficiency, and about 0.2% of those have complete deficiency. And to this end, this knowledge could have quite an impact on a large portion of the patient population. We get asked the question a lot, how many people could actually benefit from preemptive PGX? It's been estimated by Dr. Aquilante and others that as much as 99% of the population would carry an actionable PGX variant, and at least one of 5 key PGX genes listed here. The next slide, well, so let me give you an example here, sorry. So if we just look at CYPC19, sorry, 57% of our current Biobank participants would not be CYP2C19 normal metabolizers. 15% of the UC Health patient population is on at least one medication affected by CYP2C19. And if we actually scale to 500,000 Biobank participants, you could estimate that over 42,000 individuals for which we would have over 42,000 individuals in whom CYP2C19 genetic information could potentially be useful in the clinical space. I mentioned earlier about how we stack up Biobank compared to other programs. This slide illustrates that if we think about all of the research biobanks out there to date with more than 60,000 samples and who are clinically returning PGX results, CCPM stands out amongst all of them. And this preemptive clinical PGX via a research biobank is particularly feasible in the setting of strong institutional support, which we have, genotyping in a CLIA environment, which we have, a multidisciplinary implementation team, and I can't underscore that enough in CCPM, and also proactive strategies to engage stakeholders. We're very proud of this manuscript that was led by Dr. Aquilante and Keo last year, which still stands as a landmark paper as a how to build such a program at your institution. It's important to underscore increasingly that we're made aware of the tremendous genetic diversity across ancestral groups. And this slide is just a reminder of really not until very recently, just a little over 2 years ago, did one of these consortium for which we rely on making decisions about selecting drug gene pairs really begin to expand how they define various ethnic and racial groups, now relying on the 1,000 Genomes Project, phase 3, for example. And importantly, our team, as well as others, have tried to underscore the importance of this in recent years in research around genetic diversity from different populations, where we've been able to demonstrate in our own research program, for example, considerable ancestry-related bias in clinical databases, and that's used to designate clinically actionable genetic variants. And in a more recent paper that we've been able to publish, showing that as much as 10% of genetic variation has been missing in public databases specific for populations of African ancestry. And so I'll round out this part of the discussion before I hand it over to Chris to segue into this conversation around the importance of ancestry and thinking about, as I just mentioned, referring to the 1,000 Genomes Project as an example of what we know about genetic diversity. The purpose of this slide isn't for you to be able to hone in on particular numbers or allele variants, but I'm just simply trying to illustrate that for different allele frequencies for these different, in this case, cytochrome P450 genes that we've been focusing on, there can be tremendous variation in frequency of these alleles across continental populations. The example that I'm honing in on in particular here is for this particular SIP, where you see considerable more frequency of that particular allele in continental African populations compared to all of the other populations. And a better example possibly is here, to be more specific, looking at the distribution of CYP2C19 alleles across different ancestral groups. And again, not to read the detail, but what you can see for the populations of African ancestry, considerably more diversity and differences in frequency of what are typically cited as some of the most common star alleles. We tend to think of star alleles 2, 3, and 17. We would lose about 40% of potential allele calls in Africans if we only focused on those star alleles. So in other words, if a laboratory only sought those 3 alleles out of the various CYP2C19 alleles, there'd be very little benefit to the non-European populations. So at this point, I'm going to hand the podium over to Dr. Gignoux, who's going to dig deeper into this issue of the role of ancestry and what we're doing in CCPM.
Thanks so much, Kathleen. And I'm happy to talk about some of the resources that CCPM has available, some of the insights that we've gained over the past several years now, as well as our vision for some translational opportunities being developed within CCPM. And so the first thing to highlight is really that we have a large-scale data resource that is available to anyone on campus who's interested. We just wanted to highlight this screenshot from the Health Data Compass intake form, where our data is available alongside UC Health informatics data or all payer claims data. There's a slightly different process, of course, for applying, just given the sensitivities with genetic data. But we just wanted to, again, highlight that this is available. And even for people who may not feel the most comfortable with large-scale genomic research, this is a very large data set in terms of the entire sample. CCPM has put together a resource to provide additional assistance with analytical support through our translational informatics service, and that's being led by Dr. Nikita Pozdiev in the center. So, so we just wanted to highlight that across these 33,000 individuals, there's an enormous amount of phenotypic detail that we can extract out of the electronic health records. And this is some work being led by faculty Laura Wiley in the center and a couple of the analysts in her group. And what we can see is across a number of disease domains that there's, very highly prevalent traits. And so, we can start to not only think about sort of most common things, but also provide some insights into potential feasibility analyses for traits or observations in the healthcare record that may be at a lower frequency. But one of the things that we really stress in terms of biobank focused research is this opportunity to look across the entire phenome at a number of different diseases and not be limited by specific disease classes. In addition, beyond the standard kind of single genotype analyses that are available, we can use some tools in this instance from machine learning and other kind of software to predict for research things such as HLA alleles, some of the CYP alleles that Kathleen was just mentioning that that can be rather complex, but we can still derive research insights from them. And so this is an example looking at the cases and controls from our initial freeze of the CCPM biobank data, where we're able to impute or identify HLA alleles for each individual in a research context, but at least provide them for people interested in potential disease associations. And as we can see, sort of the top alleles are consistent with what's been previously reported in the literature. So like I was saying before, being able to look across the entire phenome provides some exciting insights that are really only available in a kind of resource like CCPM has. That's very rare in terms of these kind of settings across the country. One of the things that we're really interested in doing is we can take some variants that have been previously reported to be associated with specific diseases. In this case, this is a variant that was previously reported to be associated with hypothyroidism. And we can look across the entire phenome to kind of refine or identify the entire picture of associations, and kind of by that be able to unravel exactly what that variant is doing. So in our data in CCPM, not only are we able to identify associations with hypothyroidism, but even stronger associations with type 1 diabetes and associated phenotypes, as well as sleep-related disorders, in particular restless leg syndrome, which shares some etiology with autoimmune conditions. And so, in theory, we're able to do this across a wide range of variants across the genome and really paint a more nuanced picture than can be done in a sort of single variant and single disease context kind of setting. So in human genetics, and a lot of the people in CCPM come from a human genetics background, we're an extremely collaborative group, and we tend to share information where appropriate and available across a large number of different studies. And it's really with the idea that that as we gain statistical power, we're really refining our insights, and we're able to say things materially in different traits that wouldn't be possible if we just looked at analyses from a single study. And so consistent with this, there's been a lot of sharing and a lot of growth in genome-wide association studies. And so, we think about the potential for, studies in the hundreds of thousands or maybe millions of participants. But really, if we look across the entire spectrum of genome-wide association studies, hundreds of millions of people have taken part, to the point where about 1 in 500 or so individuals across the world have been part of some sort of genome-wide association study, which is a really remarkable number. It really speaks to the kind of scale of these kinds of operations and the ability to, develop tools that allow us to share insights across the globe in a lot of instances. And so, we have to think about what this all means, right? Because these tend to be very expensive studies, particularly on this kind of scale. And there's been a large number of critiques of genome-wide association studies over the years, sort of highlighting a few of these outside of the expense. Oftentimes, people find that single variants are only moderately associated with outcomes. So maybe you're thinking about an odds ratio of 1.1 or 1.2, it doesn't come anywhere close to kind of discussions of clinical actionability. As you're looking across the genome, there's an opportunity to identify genes that may be difficult to ascribe function that comes back to disease. So you see an association, and then it's, the functional work to kind of follow that up becomes extremely difficult. Oftentimes, we look at very broad phenotypes, and people who are domain experts oftentimes critique this. And there's always this trade-off of trying to understand, do you try to maximize study size and then somehow, be able to maximize statistical power versus the sort of, precise phenotype that may have stronger effects. And so there's always this trade-off. And then there's always a question of whether the phenotype being discussed is exactly the same in different kind of instances, in different kinds of contexts. But we really think that now, as we start to get to these really large sample sizes, genotype effects should really no longer be considered weak. And what we mean by this is that really the insights that can be gained really benefit from summing up all of these small effects across the genome. And so maybe as, even if a single variant has a effect size of 1.1 or 1.2, once you start aggregating all these effects across the genome, potentially hundreds, thousands, even potentially millions of variants impacting a trait, you're starting to see effect sizes that can be very large. Let's say on the order of an odds ratio of 3, 5, maybe even 10, depending on the disease and sort of where you set a threshold. And so this is just an example from a more theoretical paper now that's almost 10 years old, but it kind of provides some guidance on how relatively small genome-wide association studies actually tell you something meaningfully different from the kind of insights that we're extracting now from studies on the order of hundreds of thousands to millions of individuals. So now we're really able to get those kind of weak effects across the entire genome, but measured precisely because their sample sizes are very large. And so we're actually able to calculate these kind of risk profiles. So here's an example from a high-profile paper from a few years ago, demonstrating that, that for coronary artery disease in this instance, and this is real data, this, the curve may look sort of idealized, but it's based on real data of about 400,000 individuals from the UK. And you can identify that there's individuals walking around with extremely inflated risk or higher risk of coronary artery disease than the general population. And so this is just one example. One of the things that's interesting here is that they were kind of describing that this information provides statistical support above and beyond things like family history, carrying sort of potential pathogenic mutations, that there's additional clinical utility to this kind of data. And this was really exciting to the field. And, we can think about scaling this up because with a genome-wide platform like we have in CCPM, we're not limited to a single polygenic score. We could calculate this for any number of traits. There's a huge caveat to this kind of application. And going back to what Kathleen was discussing before, we need to look at actually who is contributing to these genome-wide association studies. So if we actually look at this information, what we see is across time that populations of European descent have been overrepresented in these kinds of data sets compared to other groups around the world. While this has been pointed out multiple times in the human genetics literature, it continues to remain a challenge. And as large-scale data sets from countries across Europe are coming online, this pattern is likely to continue for a very long time, unfortunately. So let's actually see what happens when we sort of take some of these discoveries, particularly discoveries in European populations, and then apply them in other groups using the same kind of polygenic scoring as was done in the coronary artery disease example. And so here's a highly heritable trait, height. It's very easy to measure. We have it measured in a lot of people. And so our polygenic scores are relatively good. But what you see when you apply it across populations is that the distributions of these populations shift around. Now, within each of these groups, one would generally assume that these polygenic scores still have some predictive accuracy. But if you take all these groups together, what you see above all else is the impact of genetic ancestry or population structure. So a naive application here would make people assume that East Asians or Africans may be like 3 feet shorter than Europeans on average, and certainly that's not the case. And so really what we need to do to understand how these risk factors actually play out is look at both ancestry and these polygenic scores in context. So it doesn't just matter what someone's score is, it matters what their score is compared to a group of individuals who come from a similar genetic ancestry background. And so this really affects interpretation and sort of describes why it's so critical to understand both ancestry and risk in context of each other. And in CCPM, as is common in most large metropolitan biobanks, we have a number of participants with ancestry from all over the world. Here's an example of 500 individuals with this kind of structure analysis run by Jonathan Short, an instructor in the division. What we can see here, each tick on the x-axis represents one of these 500 randomly selected individuals, and the colors represent their ancestry from all over the world. Now, what's important to note here is that that these clusters don't necessarily neatly fall under sort of OMB type categories. What we're really seeing across individuals is a sort of continuous spectrum of genetic ancestry. And that's what we need to know in order to appropriately calibrate risk for individuals in this kind of polygenic context. So one of the things that we've been really exciting to develop along with colleagues is a sort of large scale attempt to kind of counteract this European bias by, again, combining forces with a number of colleagues, both nationally and internationally. Now this has resulted in a couple of funded R01s to specifically work on this stuff. In particular, we're really excited about an opportunity that we're co-leading alongside collaborators at UNC and the Fred Hutch Cancer Center to look at cardiovascular risk across a number of different populations from around the world, and really focus on doing a better job with methods of estimating this risk in a manner that is appropriate for individuals of very diverse backgrounds. And so the idea then is, you know, with these kind of insights in place, ultimately, we'll have a much better opportunity to provide these kinds of results in the future for anyone who walks into the clinic, right? Not just people of a certain ancestral background. And just as an example of this kind of thing, this is some work being led by Michael Rosenberg, who's a joint faculty with us and cardiology. He's an electrophysiologist. And so we've been looking at atrial fibrillation across populations within CCPM. What you can see again on the left is this kind of naive implementation, not thinking about ancestry at all. And we can see this kind of bias in distribution that happens between different groups. You know, when we're sort of aware of this, we can kind of recenter everyone. And so we can provide a more nuanced description of this kind of ancestry, and just to ensure that, like, you know, these are actually potentially of clinical interest. Ultimately, you know, a very simple kind of predictive model, including age and sex, we get a AUC above 80%, which could potentially have additional clinical insights that we're following up on. So, of course, like a lot of people, both on campus and around the world, we've wanted to be responsive to the ongoing pandemic with COVID-19. Again, like I was saying before, given human genetics sort of culture of collaboration, a group in Finland started to organize a large-scale host genetics initiative, a genome-wide association study of a large number of different COVID-related phenotypes. Just wanted to encourage people to take a look. There's an interactive browser that's available today. People are welcome to go up and look. The summary statistics are publicly available. And we were able to contribute as one of 4 EHR-linked US biobanks out of the roughly 50 total studies that have taken part in this. So we're really excited to continue working with this and identify new potential collaborations with groups both here and abroad around addressing COVID-19. So with that, we've developed in CCPM a large-scale resource, including both informatics and genomics data, to look at potential risk factors and other things that, that we can help interrogate and study. This is a highly collaborative effort involving roughly 15 people within CCPM, looking at different hypotheses. But in our latest data freeze, this includes over 30,000 COVID positive individuals. So looking at potential associations with severity and that sort of thing are quite possible in a data set like this. And includes an extremely rich set of informatics data if people really want to drill in on specific hypotheses. And just to give an example of what we've been able to do, this was some work led by one of the people in that translational informatics services that I was talking about before, where we can actually look at the degree to which individuals who test positive for COVID-19 and end up being hospitalized are coming in with specific additional risk factors. And so, you know, being able to sort of look at this across the spectrum of different racial or ethnic groups, we think is really critical to understanding both the disease process in general, you know, who's being treated, who's waiting longer before they come in, that sort of thing for surveillance. So with that, just wanted to hand it back to Kathleen for some last thoughts.
Yeah, thank you, Chris. We're looking forward to any questions you have. Before we move to that, the two of us want to really shout out to the many faculty and staff at CCPM that made all of the information we've shared with you today possible. Special shout outs, as you heard, for Laura Wiley, Jonathan Short, Christy Crooks, who runs the Biobank, Nick Raphaels, and Samir Shavan. Really important to note, at least for the first half of this presentation, the many PICLO members, both past and present, who made our PGX initiative and its launch possible. We absolutely couldn't have come as far as we have in the first 5 years without UC Health leadership. Especially want to thank Steve Hess and CT Lin for all they've done to help us put these pieces into place. And importantly, the UC Health IT service lines. If you want to learn more about our PGX initiative, we'll leave up here both Chris and Dave's contact information. We've also added information on TIS, our Translational Informatics Services, if you want to learn more about how you can access and leverage CCPM data in your own initiatives, as well as Health Data Compass now under the directorship of Dr. Ian Brooks. So thanks for your attention. And again, we look forward to the questions.
Okay, well, that was a great presentation. Really enjoyed it. And it's amazing what you've been able to build over a short period of time. So congratulations. I've always wanted to ask population geneticists this question. So since I have two of them on the line, I'll ask both of you for your opinion. When you see differences in allele frequencies across different ancestries, what does that tell you about environmental stress? And how large those differences have to be between populations to focus you in on that particular question? I would imagine that there are quantitative differences that would speak to environment influencing the genome, and there are differences that would suggest that it might be due to random variation. I'd love your opinions on that.
Chris, you should take that.
Yeah, it's a good question. So so there are certainly statistical tools that allow you to tease those two different sort of processes apart. You know, usually a lot of what we see in humans just across the world is or tends to be random. So that's sort of the kind of sort of benchmark to test against. Oftentimes people call it genetic drift. And so, then the question becomes, is there a sort of surprising change in allele frequencies that you wouldn't see just due to that random chance? Those are the kind of tests that people look at. And there's certainly some cases where we kind of know that processes have happened in the past and humans have undergone selection, and in other situations it can be very hard to ascribe a specific environmental sort of pressure in that kind of instance. But yeah, the situations where you have things like lactase persistence and sort of adaptation to infectious disease, like the Duffy null allele, those kind of things tend to be relatively rare in the genome.
Thank you. Kathleen, any other comments?
No, I think he hit it.
Okay, good. So let me ask a very practical question, because I think that there's quite a bit of interest in the translational informatics services. How does one, A, go about accessing that? And B, can you learn about a specific genotype that may be of interest to an investigator? How specific can you get about the phenotypes that people are interested in? Is this diabetes in general, or very specific types of diabetes, hypertension, cardiovascular disease, et cetera?
I'll start that, and Chris, feel free to jump in. So, just to let everybody know, typically, or until very recently, for the phenotype questions, David, you would come directly to Health Data Compass. You would be assigned an individual within Compass to sort of shepherd you through that process, help you design your study and ask the questions you want to ask of Compass. Again, till recently, strictly around phenotype data, if you will. We've now added all of the genomic data that we've generated into Compass. And so it's a combined data set. We formed TIS, the Translational Informatics Services, just in the last 9 months to be able to really bring together that search, for example, through the tool Eureka, if you will, in Compass, and to facilitate that, make it a little more user-friendly with researchers. And then Chris also mentioned the relatively newly formed committee that we've created, ABC, which is, if you, David Schwartz, want to ask a question that includes both phenotype and genotype data, it's really a partnership between TIS and Compass. Compass is sort of your gateway into that, and then TIS will provide those sort of hand-holding services if you need it. You will then take your proposal to the ABC committee, who's simply there to check the boxes. You have IRB approval to ask the question that you want to ask, and so on and so forth, to give you access to those data. So, Chris, do you want to add anything to that?
Yeah, just to say, where people might be interested in using CCPM data, let's say in a grant or something, there's some opportunities, and it's worth checking with the TIS individuals about sort of a quick way of identifying a particular cohort size. So if you're interested in potentially 300 people or 3,000 people as part of the study, and also and with that we are a true biobank, so these samples are stored if there's a particular question that requires additional data generation, and that gets approved through this ABC committee, then there are samples that would be available for downstream research.
Mm-hmm. And I do want to shout out to Chris and his team already. They've done quite a bit of sort of off-the-shelf genomics, if you will. So for example, generated GWAS data on 5 traits, 5 relatively common traits. As you saw from Chris's side of the presentation, quite a bit of work around FEWAS. And so as much as we can, we're also trying to generate in CCPM the sort of off-the-shelf preemptive genomic data that's available to the research community.
Maybe just to clarify to the audience, Kathleen, tell everyone what FEWAS is.
So we really turned GWAS on its head. So instead of looking at, in the case of the mega, 1.8 million SNPs and then imputing another 6 million for a single trait, you're now flipping that over and saying for a particular SNP or variant that I know has been strongly associated, the example Chris gave was hypothyroidism, how, in what other traits is that variant also associated?
Okay. And you can look across the entire spectrum of traits that are in the health data compass, essentially.
Exactly.
Yeah.
Exactly.
Yep.
One question is what's your threshold or what's your process for deciding to make a recommendation for a particular gene variant medication outcome versus holding back and waiting to acquire more data? How is that decided in your group and outside of the group? What are the benchmarks to make those recommendations?
Yeah, that's a great question. So this falls under the purview of Piccolo, and within Piccolo, there's actually a working group that makes those decisions. And what they do is refer to, I mentioned those national organizations, particularly CPIC guidelines. They're really divided up or tiered, if you will, as CPIC A, B, and so on. CPIC A guideline or recommendations would be for those drug gene pairs that are tried and true, compelling evidence that there's an association between a particular allele and response to a drug. As you go further down, and I'm constantly educated by Chris Aquilante on this, so I appreciate that, Chris. I'm an asthma geneticist. I'd love to see some asthma drug gene pairs in there. They're way down the pipeline right now around CPIC C or D, not recommended for return of results. So we maintain a very strict threshold within CCPM of really focusing on the CPIC A guidelines. But this working group within Piccolo is constantly reevaluating updates from these organizations and modifying as we can.
Good. So how have you achieved such good representation of Black, Indigenous, people of color in your biobank, and what hurdles have you had to overcome in terms of trust and community outreach?
We're trying to do a lot better. We know we can do a lot better than we have, even though, reflecting on that image, we're better than other sites. We know, for example, that within the Metro Denver area, some 15% of the population is of African ancestry, but we're not seeing that representation in our biobank. A population that we are particularly keen to expand recruitment is the rural population, and we have tremendous opportunity at this institution to do that. I think, David, creating what have we done well? I think creating a relatively seamless, relatively easy consent process along with the video, and we've got a new video we're about to roll out that is even more, I think, dynamic and inclusive, has been a big boon for us. We have our patient community engagement committee that we work very closely with for recommendations. A major initiative for UC Health is to work with us to broaden that diversity, however. So there's a lot of work yet to be done.
Good. One last question. When you consider genetic ancestry, or have you had the opportunity to consider genetic ancestry in assessing COVID risk in various populations. Risk for disease and risk for severity of disease, since the distribution of disease is so different across different ancestries.
Chris, you want to take that one?
Sure. Yeah, I mean, I'm sure the audience here is very familiar with the ongoing extreme disparities, both in susceptibility and severity of disease. So, you know, when it comes to questions like that, it's you know, it's definitely something on people's minds, and we have, I think, some approaches that can kind of appropriately ask that question. We certainly want to be mindful of not, you know, trying not to conflate genetic ancestry with other aspects that that are different in different racial or ethnic groups, and certainly you know, differences in environment and socioeconomic status and that kind of thing really are such big factors in COVID. So, I mean, we're certainly interested in it, and where we can kind of provide insights, it's definitely something that we're interested in doing, but very carefully.
Mm-hmm. Well, this has been a really great discussion. I think importantly, the resource that you've created is something that should be of keen interest to a lot of our faculty and trainees, and I'm sure you'll be hearing from a lot of people in terms of how they could potentially collaborate with you and work on really interesting projects, important projects, in terms of understanding the distribution of disease in our population and also the way in which we can impact the disease. So thanks so much for preparing this talk and giving it. Do you have any final comments you'd like to make?
David, we just really appreciate the opportunity to share with our research community and clinician scientists across the institution what we've built. We need physician champions. We need all of you to advocate for this initiative and get the word out. So thanks for everybody who attended.
Okay, great. Thanks very much.
Take care.
Thanks.